Welcome everyone to the session and thank you for joining us. The live transcript is available if required. If you prefer to hide it, just click on the live transcript button and select Hide Subtitles. The session will be recorded and the link will be sent to you afterwards, and it will be available to view for 7 days. Of course, it's available at all times for members of the society. Please use the Q&A function if you have any questions you'd like Dr. Ryan Hunter to answer during at the end. I will post those questions to him. Feel free to use the chat to introduce yourselves and for general comments and suggestions for future topics. Allow me to introduce our speaker, none other than Dr. Ryan Hunter. Dr. Hunter hails from Canada. He was born in Toronto. He pursued his bachelor's and PhD studies at the University of Guelph under the guidance of Terry Beveridge before doing his postdoctoral research, which he will expand shortly. I met Ryan, whom I insist on calling him Rick for all these years, at the biennial Pseudomonas conference in Hanover, Germany in 2009. Here is an example of why in-person conferences are important, and it provides the opportunity to network. Our chance meeting at the conference led to many meals and continuous interaction over the many years at the various conferences. He started as a faculty in University of Minnesota in 2013 and has authored more than 50 papers. And I invited him to join the JMM editorial board in 2017. Now he has risen to the position of senior editor for the microbiome and microbiology ecology sections. So think about attending meetings and networking. They're such a crucial part of your career. His talk is titled, When Good Bugs Go Wild, the Role of Commensal Microbiota in Chronic Airway Diseases. So thank you, Ryan. Take it away, please.
Thanks, Kalai. Excuse me. And thanks all of you for tuning in this morning or this afternoon, depending on where you are. Today, I am, well, I'll start out by giving a little more information about my background and where I received my training and how I arrived at where I am today, including my lab's research. But then I'll take a deeper dive into some emerging data that's coming out of ongoing projects in my lab, focusing on our commensal microbiota. So these group of organisms are traditionally not really considered to be pathogenic or contributing to health and disease in many ways. But we have a lot of emerging evidence to suggest that this group of organisms plays an important role, not only in the onset, but also progression and potential treatment of chronic airway diseases as well. So I'll present some of the latest and greatest and newer projects that we're excited about in the lab at this time. So just building on what Kalai mentioned in her introduction, I am from Canada, Ontario to be specific, and I spent 11 years of my education at the University of Guelph, including my undergraduate and doctoral studies. At the time, as fresh out of high school, I didn't really know what I wanted to do for my career. I started out in nutritional sciences, then I moved over to engineering. But it wasn't until my third year where I really hit my stride, and that's because I took a course with Terry Beveridge, and he's shown here on the right. And Terry was not only a renowned microbiologist, but he was a super talented microscopist. And as an undergraduate student, he let me use his electron microscopes to peer down into the phosphorescent screen. At that time, we didn't have computer monitors attached to electron microscopes, so I did have to look at the screen. And he just opened my eyes to the beauty of the microbial world. So, and these are some images that he either he took or helped me take. And he let me visualize, the S layers on archaea, the mineralization of gold and the intracellular space of other bacteria, and also the accumulation and precipitation of heavy and trace metals on bacterial cell surfaces. And this was sort of the focus of my undergraduate work. But then I stayed around to pursue my doctoral studies with Terry as well. And he started to encourage me to think about how environmental conditions matter to the behavior of the microbes that we're interested in, and how they behave in situ in the natural environments in which they're found. And this can have a pretty profound effect on their physiology, which we now know. So for example, and if you take a soil environment, like I'm showing you here on the slide, if you stick an oxygen electrode into the ground, just a couple of hundred microns below the surface, oxygen becomes limiting. And so what this does, it forces microbes to adopt all kinds of versatile and fascinating modes of metabolism to thrive, which in the environments in which we find them. So this can include, fermentation, methanogenesis, the oxidation and reduction of nitrogen and sulfur compounds, but also the redox transformation of trace and heavy metals as well. So this can include iron, manganese, and all kinds of other elements that we find in the soil environment. And as I said, this can have a pretty profound effect on the behavior of the organisms that we find there. So even just in this electron micrograph that we see here, you can see that there's a lot of submicron heterogeneity, and this is all driven by the local environmental conditions that surround these diverse bacterial cells. This can have a pretty far-reaching impact on the bulk macro environment in which these bacteria are living. But we can also manipulate these types of interactions to our advantage, in this case through a bioremediative lens as well. And so this comprised the bulk of my PhD, thinking about how microbes behave in the soil environment and how this impacts many of the metal and redox transformations that they undergo. From there, I wanted to build on my ecological skill set, and I really wanted to learn bacterial genetics. And so for that reason, I reached out to Diane Newman, expressing interest in pursuing my postdoctoral studies with her, first at MIT and then at Caltech. And she, well, backstory, Terry Beveridge was also Diane Newman's co-mentor for her PhD studies. And so that was a common connection there. And so it sort of opened the door for me to join Diane's lab for my postdoc. And so she helped me learn bacterial genetics, mainly in the context of Pseudomonas aeruginosa. But Diane also helped me realize that many of the same questions that I was asking during my doctoral studies about natural environments, so for example, the soil or water column, we can also ask about bacterial pathogens in vivo in the context of human disease. So likewise, in that same scenario of the soil environment I showed you on a couple slides ago, if you stick an oxygen electrode into a mucus layer, for example, we also know, based on studies that came from Diane's lab, that oxygen also becomes limiting. So just a couple hundred microns below the surface, oxygen becomes depleted. And once again, this plays an important role in dictating which organisms can thrive there. So we know that we know a great deal at this point about our microbiota and bacteria that live on and within us. But I would argue that by comparison, we actually have very little understanding about the in situ chemical environments to which these bacteria are exposed. How do these environments impact how they're behaving on and within us? And how does this contribute to health and disease? And so I started to think about this during my postdoc, and really in the 10 years since I've been here at Minnesota, this overarching research question of how do environmental microbe interactions impact health and disease has essentially become the primary focus of my lab's research. And so we ask questions like, how do environmental conditions impact both the composition and behavior of host-associated bacterial communities? How do these processes or environmental microbe interactions contribute to disease states? How do these conditions impact some of the antimicrobial treatments that we use? And then ultimately, can we manipulate these sorts of environmental microbe interactions to our advantage to perhaps better curb the onset of infection, but also treat infections after they have started? So we're interested in all kinds of sites of infection, but traditionally we've used the cystic fibrosis airways as our model system. So cystic fibrosis, or CF for short, is an autosomal recessive genetic disorder, and this affects the transport of ions across cell membranes. It's a multi-organ disease. It can also impact the pancreas, vas deferens, gut, liver, and sinus cavity as well. But it's perhaps best known, or the most well-known manifestation of the disease is in the lower airways, where you get increased thickness and viscosity of the mucus layer due to dehydration and reduced mucociliary function. And so this leads to chronic bacterial infection over time. And ultimately, this results in the bulk of mortality in the majority of the CF population. We know a great deal about CF microbiology, and this has been based on 4 or 5 decades now of clinical culture data. So what I'm showing here, this slide is produced by the Cystic Fibrosis Foundation, but on the y-axis, I'm showing you the percentage of individuals with the disease that are colonized with a traditional pathogen. So this begins shortly after birth, where the majority of patients are colonized by Staphylococcus aureus, and that's shown by this green line here. But as patients age and they reach adolescence or even adulthood, you can see this blue line start to increase. And this represents Pseudomonas aeruginosa. And it is viewed as sort of the canonical pathogen of the CF airways. It's considered to be a major player in airway pathogenesis. And so the detection and treatment of CF disease is largely focused on clinical culture of this small set of organisms associated with worse patient outcomes. But this is really met with mixed success. And so there's a hope that we can improve upon our detection of these organisms and better understand the ecology of the airways. More recently, thanks to the advent of culture-independent sequencing, we now know that the CF airways are more complex than previously appreciated in terms of their microbial community composition. And that's represented by these sequence data here. It's just a subset of data that's been generated by my lab over the past decade. And all I wanted to point out, don't get wrapped up in the colors, but just in addition to many of these organisms that I showed you on the previous slide that are viewed as the traditional set of pathogens, what these data tell us is that the lungs are colonized by a number of other bacterial genera that are not really traditionally viewed as pathogenic. We're interested in many of them, including Fusobacterium, Streptococcus, Veillonella prevotella. These all are typically associated with the oral cavity. But whether and how they play a role in CF disease progression is poorly understood. So one of the major areas of emphasis of my lab is to really understand the specific contributions of many of these non-traditional taxa to disease, but also how interactions between them and the host shape CF lung infection. So our entry into this problem came about a decade ago when I started my lab here in Minnesota. And one of the primary questions that we wanted to start out with was the nutritional environment of the lung. What nutrient sources sustain these dense bacterial populations that we find within the airways? So in the lung, Pseudomonas, for example, can reach about 10 to the 7 to 10 to the 9 cells per gram of mucus, which is a highly dense bacterial community, rivals what you might see in the gastrointestinal tract, for example. But in contrast to the GI tract, when we dove into the literature, we were pretty surprised with how little was actually known about the in vivo nutritional preferences of Pseudomonas at the site of infection. Unlike the gut, where you have dietary nutrients coming in all the time, and where a lot is known about bacterial nutrient utilization, that doesn't really happen in the lung. There's no dietary nutrients coming in. So anything that these bacteria are using intuitively has to be host derived. And so that's why we focused on mucus itself, or mucins in particular. So these are the main macromolecular constituents of mucus. They're megadalton-sized glycoproteins, and they consist of this polypeptide backbone that I'm showing here in green. And this polypeptide is heavily decorated by O-linked and N-linked glycans that comprise about 90% of the dry weight of these mucin polymers. They're incredibly carbon dense and represent a rich source of nutrients that bacteria can potentially tap into. Mucins are widely recognized to serve as a nutrient source in the gut microbiology literature. But when we dove into the literature, we found that very little was known about whether Pseudomonas itself can use mucus as a carbon source. And if so, what molecular strategies this particular bacterium uses to do so. And that's sort of the first question that we wanted to tackle in my lab once I started here in Minnesota. So coming from a genetics and physiology background, we wanted to use a forward genetics approach to try and tackle this question. And so to do so, we use an approach known as TN-seq, or transposon sequencing. If you're not familiar, the way it works is you generate a mutant library of your given bacterium of interest. In this case, it's Pseudomonas aeruginosa. And each cell within that library contains a transposon insertion in a given gene. You pool all of those cells together in a high-density transposon mutant library, and then you grow it out under a defined set of conditions. In this case, we're growing it on mucin as a sole carbon source, and we compare it to just growth on glucose as a minimal medium. After outgrowth, after an X number of generations, you then use high throughput sequencing. In this case, we used Illumina, and you map the insertion sites of each transposon in the library that results from your experiment. And essentially, this take a step back. If you sequence this pool of transposon insertion sequences, if a gene in which a given transposon is inserted falls out of the population, this might tell you that that particular gene is essential or at least important under that defined set of conditions. Essentially, it allows you to come up with a quantitative fitness score for every non-essential gene in the genome and its essentiality for growth under a defined set of conditions. And in this case, it's mucin as a sole carbon source. This is where we started running into our first problem. In order to do a TNSeq experiment, you generally need your bacterium to grow to allow some of the mutants to fall out of the population. But when we created a defined medium in which mucin served as the sole carbon source, in this case, it was 15 grams per liter, so pretty carbon rich, Pseudomonas only got up to an optical density of about 0.1. And this is actually quite poor for Pseudomonas, who can grow on almost anything, including water. And so this is frustrating because we needed it to get to a fairly high density and to allow us to do our TNSeq experiment. And so we decided to artificially increase the mucin concentration to a non-physiologically relevant condition of 30 grams per liter. And in this case, we started to get some growth. So even though it was somewhat contrived, we reasoned that any molecular insights we could gain about Pseudomonas and its ability to use this glycoprotein as a carbon source would be informative and help us better understand how it might be behaving in vivo. Under these conditions, Pseudomonas adopts a biphasic growth phenotype. It's somewhat, maybe a little bit difficult to see on the log scale here, so I'm just going to show it on a linear scale. And we characterize this as our Pseudomonas biphasic growth pattern, where you have a first phase where they get up to an OD of about 0.3, and then it appears they get up to an optical density of about 0.8. So this suggests somewhat of a hierarchical carbon utilization scheme as it's growing in this medium. On the right, I'm showing our experimental approach. And so we wanted to know how Pseudomonas might be deriving nutrients in these two phases of growth. So we took our transposon library of 70,000 mutants. We saved one aliquot as just our reference library. We then took another aliquot and grew it up to an optical density of 0.3. So this represents that first phase of growth. And then we took the spent medium from this experiment in panel C here, and use that as the growth medium for a second experiment in which this first phase carbon source had been depleted from the medium. So this gives us an idea of what they might be using during this second phase of growth. We then captured all of the outgrowth, performed our PCR enrichment and transposon sequencing, mapped all those sequences back to the genome, and again came up with a quantitative fitness score for every non-essential gene within the Pseudomonas genome under these conditions. In that first phase of growth, we found a total of 68 genes that were required for Pseudomonas growth on mucins, including 30, some of which I'm showing here, that are involved in central metabolism. Among them, multiple genes shown here were required for growth in that first phase that were not required for growth in the second. So this gave us an idea of what nutrient sources they were using during this early phase of growth. Two genes of interest were ACEA, encoding isocitrate lyase, and GLUC-B, which encodes malate synthase. And these are genes encoding proteins that are involved in the glyoxylate shunt. And so what this might suggest is that, that by knocking out these two genes, it induces a fitness defect in Pseudomonas because they're not able to use either a 2-carbon compound, for example, acetate, or fatty acids. So these are both substrates that require the use of the glyoxylate shunt when they're present as a carbon source for Pseudomonas. We also found a number of genes involved in amino acid biosynthetic machinery. So what this suggests is that although Pseudomonas was growing on a carbon and amino-rich carbon source, that the liberation of amino acids from that glycoprotein were not sufficient to satisfy its nutritional growth requirements, at least in the second phase of growth. Interestingly, in the second phase, we didn't find any genes that were required for the second phase that weren't required for the first. So all of these amino acid biosynthetic machinery genes go away. They no longer require the glyoxylate shunt. And so what this suggests is that they are then able to acquire nutrients that it needs to grow. It might also suggest that nutrients are being shared between mutants in trans. So just because you have a transposon insertion in a given gene or pathway, intermediate or other metabolite might be provided by another member of that population, allowing that population to grow. So we hypothesized that under this second phase, they were potentially using amino acids as their carbon source, allowing them to get to that higher OD. We performed some additional experiments to validate this model. So we measured their nutritional substrates in the growth medium. So in black, I'm showing their biphasic growth pattern. In red on this first panel, I'm showing the availability of acetate in the growth medium. So even though it's not present at the beginning of growth, you do see this increase shortly after it grows, but then a depletion from the growth medium over time. So what we interpret this as meaning is they're liberating acetylated sugars from the mucin glycoprotein and then consuming them once they require acetate as a nutrient source. And then we measure total protein in the growth medium as well. And we can see that during the first phase, there doesn't appear to be any change in total protein available. But once they reach that second phase of growth, they are depleting protein from the growth medium. So it does support our working model of nutrient consumption. And then being a geneticist, we love creating mutants in the lab. So once again, we knocked out that ACEA gene, which is that first step in the glyoxylate shunt. And you can see that right from the beginning, it's defective in growth, suggesting that acetate is required or used from these mucin glycoproteins during growth. And then we went through and knocked out a number of proteases that Pseudomonas encodes, including LAS-B, which is elastase B, and APRA, which is alkaline protease. And while it seems to grow fairly well just in that first phase of growth, they are defective in the second phase, suggesting that they are gleaning some protein or amino acids from the mucin glycoprotein backbone. So this led to our initial working model of nutrient consumption by Pseudomonas when mucin is present as a nutrient source. Our interpretation is that Pseudomonas is able to use its repertoire of extracellular proteases to degrade the non-glycosylated regions of mucin or apomucin. And they're able to derive acetate from acetylated sugars that decorate these O-link glycans, including sialic acids, glucanac, and galnac. But once these nutrient sources are depleted from the medium, Pseudomonas is no longer able to grow, and that might explain why growth on this glycoprotein as a sole source of nutrients is limited when we provide it under a physiologically relevant condition. But I want to get back to the slide I showed you earlier and talk about just how Pseudomonas growth is inefficient on this nutrient source. This was a project that was started by a postdoc, Jeffrey Flynn, in my lab back in 2013. And he wanted to characterize Pseudomonas growth on mucin even further. So these, it's a separate experiment, but I'm showing you a similar data that if we create a defined nutrient medium in which mucin serves as a sole carbon source, and in this case, we're using MUC5B, which is one of the primary mucins found within the airways, Pseudomonas still grows very poorly and only gets up to an optical density of about 0.1. This is the molar equivalent of about 65 millimolar glucose. So again, this is incredibly carbon rich. So it was somewhat surprising that Pseudomonas was not able to grow to an appreciable density. Keep in mind, if you grow Pseudomonas in just 1/10 the concentration of carbon in the form of a simple carbohydrate like glucose, it does just fine. So it really speaks to the inefficiency of Pseudomonas to grow on this nutrient-rich compound that's so abundant within the airways or the environment in which it's found in the body. So this is frustrating because this is kind of how we wanted to start out the lab's research and really start to manipulate the ways in which Pseudomonas could use mucus. But we took a step back and started to consider we started to think more ecologically about the CF airways and what might be affecting Pseudomonas behavior. So drawing on my ecological background, we started to think about microbial interactions in the actual in situ environment in which Pseudomonas is living. And we started to think about its friends, the other bacteria that were also living within the lower airways. Once again, we are very interested in Fusobacterium, Streptococcus, Veillonella, Prevotella. I remind you that these are all typically associated with the oral cavity, and in the mouth, they're all fairly well characterized for their ability to degrade salivary mucins as a nutrient source. In doing so, not only can these bacteria grow on their own, but they can also liberate secondary metabolites like peptides and amino acids, TCA cycle intermediates, fermentation metabolites like short chain fatty acids, and also cysteines as a source of sulfur. And this allows secondary colonizers within the oral cavity to colonize, and this is what helps structure very complex, dynamic bacterial communities that reside in our mouths. So we started to think that, maybe Pseudomonas doesn't do it on its own. These bacteria are regularly aspirated into our airways. If you have a healthy innate immune system, you can either clear those anaerobic bacteria by mucociliary clearance, or they're removed by intact antimicrobial peptides, for example. But if you have a genetic defect where you can't clear those organisms as efficiently, then their residence time might increase, and they might help create this nutrient pool that Pseudomonas and potentially other pathogens could then capitalize on. And so that became our next working hypothesis. And we asked the question is whether or not these commensal microbiota, which we also refer to as anaerobic bacteria, I'm going to use that interchangeably throughout the talk. The question is, do these anaerobes support the growth of Pseudomonas via mucin degradation? And might this be happening in vivo? So my postdoc, Jeff, was very creative. And so one day he just decided to walk around the lab and ask his lab mates to start spitting in tubes. He wanted to culture their oral microbiota and use these as our model anaerobic communities to test whether they might be able to stimulate the growth of Pseudomonas, but also shape its behavior in the presence of mucin as a sole carbon source. So this is actually an experiment with Jeff's oral community. When he used his saliva as the inoculum under anaerobic conditions, whatever was growing in his mouth grew just fine. It grew up to an optical density of about 0.7 or 0.8. So something is able to grow on our defined mucin medium, just not Pseudomonas and some of the other pathogens that we had tested. Just out of curiosity, we sequenced what came out the other end of Jeff's growth experiment. And sure enough, it was those four bacteria that we were particularly interested in: Fusobacterium, Streptococcus, Vionella, and Prevotella, and then a number of other genera that were present in smaller percentages. So this supported our hypothesis that oral flora could potentially break down airway mucin and potentially cross-feed other pathogens in vivo. But it also gave us a tool in the form of a semi-defined airway-relevant anaerobic community to test that hypothesis in further detail. So Jeff came up with this very simple cross-feeding experiment that we now use very frequently in the lab to look at interactions between anaerobic bacteria and aerobes without the use of an anaerobic chamber. So Jeff just took his defined community and put it in the bottom of this culture tube, just a normal glass culture tube, in the presence of 10 grams per liter of mucin and a 1% agar gel. So this is meant to mimic like a viscous sputum environment. Once that agar gel polymerizes, over top, we put Pseudomonas in a 0.7% agar gel with no nutrient source. So under these conditions, a number of things happen. When Pseudomonas is growing, they consume all the oxygen in the environment. So this creates an oxygen gradient, allowing the anaerobes to grow at the bottom of the tube. But in this case, Pseudomonas is only able to grow if they're provided nutrients from the lower portion of this mixed bacterial community. And then the differences in agar concentration allows us to separate those agar plugs, and then we can plate them on nutrient agar and enumerate bacteria just to see how well each bacterial species is growing in this cross-feeding experiment. Here I'm showing the data from one of these cross-feeding experiments. On the top, on the right, you can see the characteristic bluish-green color that Pseudomonas produces in the laboratory. This is due to pyocyanin, which is one of its canonical virulence factors. And I hope you can appreciate the differences in Pseudomonas growth between tubes in which the anaerobic community was included versus those in which they were omitted. So a pretty striking difference. And if we use just plate enumeration to quantify how much Pseudomonas grows, we get about an order of magnitude increase over 72 hours relative to tubes in which those anaerobic communities were not included. So this supports our hypothesis that this particular anaerobic community can support Pseudomonas growth, but very clearly it's also promoting the production of a virulence factor that is known to be used by Pseudomonas to drive pathogenesis both in vitro and in vivo as well. We want to take it a little further and start asking questions about, okay, so crossfeeding is occurring, but what metabolites are being shared between these two groups of organisms? So the way that Jeff got at this question was he took mucus that we collected from individuals at our CF treatment center here at Minnesota, and then used some enrichment culturing. So once again, this draws on our sort of microbial ecology background that we commonly use on soil or water, just to enrich specific types of bacteria that live within our environments of interest. We take an aliquot of mucus, passage it on our defined minimal medium that contains mucin as a sole carbon source, passage it multiple times over a period of, I think this experiment was 5 days. and then use 16S sequencing to sequence the community that results. And then we also use some metabolomic profiling, both directed and untargeted metabolomics to try to identify which metabolites might be shared between community members in this enrichment culturing experiment. So here I'm showing some of the data from our enrichment experiments. So these are 12 patients which we sampled. On the top, I'm showing sequence data from their sputum bacterial communities when they were sampled. And then I'm showing the sequence data from these communities after enrichment. Don't get wrapped up again in the colors that I'm showing here. Anything in these hashed bars are anaerobic bacteria of interest, or those bacteria known to degrade mucin in the oral cavity. And what I'm showing here on the right, these are the aggregate sequencing data from all 12 patients. And I hope you can see that after enrichment, you see this increase in those mucin degrading bacteria, and we expected this going in. But I'll also point out that we did not provide an electron acceptor for Pseudomonas aeruginosa. So we're actively selecting against the pathogen, which allows us to enrich many of the other community members with which it's grown. And this is Pseudomonas here represented by the light blue. And you can see it's starting to fall out of the population during enrichment. And it really gives us a chance to hone in on the other bacteria that are present within these mucus samples. To figure out which metabolites they're producing, we took the supernatants after this enrichment culturing, and once again, we used untargeted and targeted metabolomics to figure out what they might be producing during enrichment. Not surprisingly, we found a gemisch of amino acids. So again, it's a polypeptide backbone. So likely the production of all of these amino acids by those enriched anaerobic communities are likely liberating these amino acids from the mucin polypeptide backbone, or they're producing them themselves. And then we also found millimolar concentrations of short chain fatty acids, acetate, propionate, and lactate. These are biomarkers of mixed acid fermentation that is carried out by many of these anaerobic organisms. And so this led to not only our hypothesis that these metabolites might be the ones being shared, but we also reasoned that if we could find these metabolites that are nearly exclusively produced by anaerobic microbiota, if we could find them within sputum, it might give us a biomarker or further evidence to suggest that some of these mucin degradation processes are also occurring in vivo. So then we went back to our clinic and collected I think it was 15 more sputum samples, and used targeted metabolomics to quantify acetate and propionate within these samples. Here we found somewhat lower concentrations, so only about a millimolar of acetate on average, and then only about 10 to 100 microns or micromoles of propionate on average within these samples. So this was somewhat surprising, maybe a little disappointing, but we thought that maybe a reason that propionate in particular is being kept low is because it's preferentially being used by Pseudomonas as a nutrient source. And so to get at that, we use some transcriptomic approaches to try to figure out what Pseudomonas was not only sensing, but potentially utilizing in vivo. And so we used both quantitative RT-PCR and bulk transcriptomics to try and answer that question. So in these data here, what I'm showing are two genes, ACSA, which is part of the glyoxylate shunt and specific for acetate utilization, and PRPD, which is specific for propionate utilization in Pseudomonas aeruginosa. The data shown here are under controlled in vitro conditions in the lab, in which we provide them with different carbon sources. So relative to glucose as a sole carbon source, in which both of those genes are sort of expressed at a low level, in the presence of 50 millimolar acetate, we see an increase in ACSA. Propionate, we see an increase in PRPD. Interestingly, when we include both of those carbon sources together, PRPD seems to be the one that's being expressed at a higher level. So this might suggest that propionate is preferentially used as a carbon source. So then we could go back in, collect mucus, and use targeted quantitative RT-PCR to see whether either of these genes or one is preferentially expressed in vivo. And that's exactly what we see. When we target these genes in sputum samples, we do see a relatively high increased expression of PRPD relative to ACSA. So this sort of supports our inkling that propionate might be kept low in vivo because it's being preferentially used by Pseudomonas and potentially other pathogens as a carbon source in vivo. And then we have more recent transcriptomic data suggesting that a number of genes in this operon are in the top 100 in the case of Pseudomonas in the CF airway environment. So it's indirect, but it does give us some additional evidence that propionate and potentially other short chain fatty acids are being used at the site of infection, and this may be contributing to Pseudomonas growth in vivo. Getting back to our in vitro cross-feeding model, we could test this. So we created mutants of our wild type strain PA14 that were deficient in acetate utilization, deficient in propionate utilization, and those that are deficient in both. And we tested them in this cross-feeding model. And even though when we grow these mutants on LB medium, when they grow just fine, so there's no growth defect in any of these mutant strains, when we grow them in our cross-feeding model, the either single or double mutants, we see about a 60 to 70% reduction in cell density in our cross-feeding model, suggesting that propionate and acetate are definitely being exchanged in this in vitro system. But it also suggests that other metabolites are being exchanged as well. Our working hypothesis is that this is amino acids, which are also preferentially used by Pseudomonas. And we're starting to generate more data along those lines to try and identify other metabolites that might be being exchanged, both in vitro, but also in vivo as well. One of the questions that we always get, though, is what is the role of the host? So we know, particularly in later stages of disease in the CF airways, it's a highly pro-inflammatory environment. You get neutrophil recruitment, these lyse open, they liberate their own nutritional goodies for pathogens to use, but they also produce a number of extracellular proteases, including neutrophil elastase, that can not only degrade or impact host tissue, but it can also degrade mucin itself. So the question is, might host inflammatory processes also be conditioning this nutritional environment for pathogens to take advantage of? And this was a project that was spearheaded by Sabrina Arif. She's a former technician in the lab. She's now pursuing her PhD at the University of Chicago with Ron Bleckman. But she was very interested in comparing host and microbial processes and how these might feed into the ability of Pseudomonas to use mucin or mucin-derived metabolites as their nutrient sources. So Sabrina was an expert in analytical biochemistry. So she used FPLC to characterize mucin integrity on its own, but also after treatment with both host-derived enzymes and those produced by anaerobic microbial communities as well. So mucin on FPLC chromatograms have two very characteristic peaks, including one that elutes early, and this is representative of high molecular weight mucins, and then one that comes off the column later, and this is representative of lower molecular weight mucins. And this is just the characteristic chromatogram that you might see if you take salivary mucins and run it on the column. So Sabrina took her own salivary mucin, enriched it, and then treated it with a number of different sources of enzymes. So here's the profile that you might see when mucins are treated with Jeff's anaerobic community that he enriched, that I showed you on a previous slide. You do see a reduction in the abundance of high molecular weight mucin and also lower molecular weight mucin as well. Here's the profile that you see when treated with neutrophil elastase. So this just demonstrates that when you treat with a host-derived enzyme, you also get degradation of mucin glycoproteins. And then there appears to be a combinatorial effect where you get the maximum amount of degradation of high molecular weight mucins when treated with both sets of enzymes. But interestingly, for the low molecular weight pool, neutrophil elastase actually appears not to have very much of an effect. It appears as though the anaerobic community is acting on these lower molecular weight mucins in ways that host-derived enzymes cannot. So that was all fine and well, but what Sabrina was really interested in is whether or not this matters to Pseudomonas. Does Pseudomonas view or take advantage of the resultant nutrient pools from these degradation processes differently? And that's what Pseudomonas, or what's Pseudomonas? That's what Sabrina next set out to test. And so she took the degradation byproducts of all of these conditions and then used those as basal growth medium to test how well Pseudomonas could grow on those mucin degradation metabolite castoffs. Again, this is just a different way of showing that Pseudomonas is inefficient growing on untreated mucin. So it gets up to an optical density of, again, 0.1 and 0.2. Here is Pseudomonas growth pattern when grown on neutrophil elastase degraded mucin. You can see that while it gets some bump in optical density, the total bacterial load at the end of the experiment doesn't really seem to change. So Pseudomonas doesn't really appear to gain much nutritional benefit from these host-derived mucin degradation processes. This is the growth pattern of Pseudomonas when grown on an anaerobic derived pool of nutrients. And again, you see this biphasic growth pattern. And interestingly, there doesn't appear to be a combinatorial effect when you use both of those enzymatic treatments. So this suggests that neutrophil elastase is not really contributing much to the growth phenotype of Pseudomonas when growing on these nutrient pools. Taking it one step further, Sabrina wanted to know how Pseudomonas gene expression changed when grown on these different mucin degradation substrates. And so in this experiment, she compared gene expression of Pseudomonas on mucin alone relative to neutrophil elastase treated mucin and mucins degraded by an anaerobic mucin degrading community. When we compare, this is just after 4 hours of growth, when we compare neutrophil elastase degraded mucin to the untreated control, you get differential expression of about 53 genes. This is out of about 6,000 genes in the Pseudomonas genome. When we look at gene expression on an anaerobe derived community metabolites that are derived from mucin degradation, we get differential gene expression of about 530 genes. So this represents about 10% of the genome, suggesting that this anaerobic community, perhaps not surprisingly, is driving a pretty profound shift in the overall transcriptional activity of Pseudomonas when mucin is provided as a nutrient source. These are some fairly recent data, so I'm not going to go through them all. I just want to point out some of the more exciting findings. So this is a heat map of Pseudomonas gene expression grown under those three conditions. So untreated, neutrophil elastase treated, and anaerobe treated mucins. Red means a gene is more highly expressed, where blue suggests it's downregulated. We found a number of genes involved in central metabolism, but the ones I want to point out, and this is what we're most excited about, is we see that in response to the anaerobic mucin degrading community, you see an upregulation of the NAR and NIR operons. So these are involved in nitrate reduction. We also see the upregulation of the SIO operon. So this is expressed by Pseudomonas. This is a terminal oxidase that Pseudomonas expresses that tends to be increased in expression when oxygen is limiting. We also see the expression of the ARC operon. So this is involved in arginine fermentation, which is also a means of metabolism that Pseudomonas can use when oxygen is low. So the collective expression of these genes might suggest that Pseudomonas can turn these on to better survive under oxygen-limited conditions. But the interesting thing is that when this experiment was performed, it was oxygen saturated. So even though that Pseudomonas had oxygen available to it, it seems that co-culture in the presence of some of these anaerobic mucin-degrading organisms is somehow signaling Pseudomonas to turn on these anaerobic associated metabolism genes. We don't yet know what this means. One of our working hypotheses is that this might leave Pseudomonas better primed to adapt to the oxygen or the dynamic oxygen conditions that we find within the CF airways. But in our hands, this doesn't appear to be the case. We've performed some oxygen shift experiments where we grow Pseudomonas under aerobic conditions in the presence of those mucin degradation metabolites. And then immediately upon shifting it to an oxygen restricted environment, it doesn't appear to gain any advantage in growth or survival. And so this might mean that they're just looking for other ways to dump out electrons. We're not yet sure. But these are some pretty striking data, better informing us to how Pseudomonas might be behaving in the presence of a number of those organisms that it co-colonizes with in the lower airways. All of these data that I've shown you so far sort of feed into our ongoing working model of anaerobic mediated mucin degradation and how it might contribute to the onset and progression of CF disease. All of us, shortly after birth, we have fairly pristine airways, very little bacterial load. As we age, we start to aspirate a number of organisms for our oral cavity. Most of us on this Zoom call can regularly clear those. But if you have a genetic defect, those bacteria increase their residence time, increase in abundance. They can start to condition that environment for consequent pathogen colonization, and ultimately you reach a stage of chronic infection where you get the recruitment of neutrophils, you get host inflammation, and this sets up a condition where Pseudomonas starts to take over the population and becomes the most abundant bacterium, at least in late stages of disease. A number of clinical anecdotes kind of support this working model. So I showed you this slide earlier. In early in a CF individual's life, Pseudomonas prevalence and abundance is actually very low. Remember, it really doesn't come on the scene until adolescence or adulthood. So this might suggest that early on in infection, there has to be some sort of environmental condition happening that allows Pseudomonas to eventually come in and take over. In our model, this relates to a lack of an available nutrient pool early on, but then the ultimate conditioning of a niche that Pseudomonas can then take advantage of. The other clinical antidote has to do with antimicrobial therapy. So if you're a CF clinician, you want to know what to treat a patient with, you approach them, you collect a mucus sample, send it off to a clinical lab, they take it through a panel of antimicrobial resistance testing, and they use the drug that may be predictive to work the best when administered to the patient. Sometimes that works. but oftentimes you don't get a patient response, even though the clinical lab might predict it would be effective. But it also happens on the flip side. The clinical lab might predict an antibiotic not to work, but yet when administered to the patient, they do see a resolution of symptoms and a patient response. So what this might tell you is that given isolate is not representative of the rest of the Pseudomonas community, but it also might tell you that the antibiotic that you're choosing to use might be targeting other organisms that Pseudomonas is growing with, potentially limiting the nutrient pool that it's exposed to. And so we wanted to test this in a little more detail. And we asked the question of whether this cross-feeding relationship might be targeted therapeutically. So for this, we teamed up with Will Harcombe, who's also here at the University of Minnesota. He's in the Department of Ecology and Environmental Biology. And Will is a champion of the weakest link hypothesis. And this hypothesis states is that the antibiotic susceptibility of a cooperative bacterial community is set by its most sensitive members. And so I'm going to take you through this conceptual microbial community and how this hypothesis plays out. In this mock community, we have a three-member consortium, and we provide it with lactate as a carbon source. The only problem is this second organism here, number 2, is not able to use lactate as a carbon source. Instead, it needs to rely on organism number 1 to convert that lactate into acetate, which it then can use as a carbon source to grow. The problem for organism 1 is that it's a methionine auxotroph, and so it has to rely on this second organism to produce methionine in order for organism 1 to grow. But then now the problem is that neither organism 1 or organism 2 is able to use methylamine as a nitrogen source. And so these two organisms need to rely on organism 3 to convert that methylamine into ammonia as a carbon source to allow this entire bacterial consortium to grow. So then we asked the question, well, let's test this mock 3-member community with an antibiotic. So these are actual data in a community consisting of E. coli, Salmonella enterica, and Methylobacterium extorquens. And in this condition, we use ampicillin as our antibiotic. And even though when ampicillin at quite low minimum inhibitory concentrations is quite effective at inhibiting the growth of E. coli and Salmonella enterica, in the case of Methylobacterium grows just fine. So it's highly resistant to that ampicillin. However, when they're grown under these cooperative conditions, Methylobacterium can't grow because their tolerance or susceptibility is set by its 2 partners that it's growing with within this community. So this was our mock bacterial community, but we wanted to test it under a more clinically relevant environment. So this, we went back to our cross-feeding system to see whether or not this cross-feeding relationship might impact some of the antibiotics that we're using to target Pseudomonas or its anaerobic partners under these conditions. So here I'm showing the treatment of Pseudomonas with increasing concentrations of ampicillin. I'm just showing growth relative to an untreated control. And we do this going in. Pseudomonas is highly tolerant to ampicillin. We don't see much effect of this drug on the Pseudomonas population. So this could be representative of an antibiotic-resistant strain. If we treat the anaerobic population that we enriched from my postdoc that I showed you on a previous slide with increasing concentrations of ampicillin, again, relative to an untreated control, we're able to knock that population out. So then the question is, well, let's grow them in co-culture under a condition that might be relevant to the CF airways. And here we're measuring the total amount of Pseudomonas growth when we increase concentrations of ampicillin. And here we do see a reduction of Pseudomonas bacterial load, and that's all because it's set by the susceptibility of the anaerobic bacterial population that it's growing with. Admittedly, this experiment is somewhat contrived. There's a number of other factors in the airways that's going to go into this cross-feeding relationship. But I'm presenting this because I think it encourages us to think about CF therapies through an ecological lens. Maybe we don't just need to target one organism, but if we can start understanding and manipulating metabolic interactions or other types of interactions between bacteria in vivo, we may be able to better leverage the antibiotics that we already have at our disposal to treat some of these infections, and perhaps not solely rely on clinical antibiotic testing data against a single pathogen, but rather consider the entire ecosystem and how we might be able to limit bacterial growth in these types of interactions that we're learning a lot more about within the lower airways. I'm going to shift gears just for the latter part of my talk and focus on chronic sinus disease. So for a number of reasons, we've had to pivot our research. First and foremost, because of efforts by Vertex Pharmaceuticals and the Cystic Fibrosis Foundation, new small molecule corrector therapies have come online and have really done wonders to improve the respiratory health of the CF population. So they're living longer and doing better, but they're also producing less mucus, which is great for them, but it's problematic for our research because we can no longer, or it's very problematic to study what's actually happening in vivo because it's not easily sampled anymore. But we can learn a lot about other ecosystems based on what we're already learning about in the lung, including chronic sinus disease. So in many ways, this is like the CF airways. It's an inflammatory condition lasting more than 12 weeks. And the stimulus can be allergic, microbial, but also genetic. So the CF population, nearly all individuals with CF suffer from chronic sinus infections. And one of the manifestations is cyclic inflammatory exacerbation. So they go through periods of disease flares where their sinus conditions need to be aggressively treated with antibiotics. It's also associated with the increased production and stagnation of mucus within the upper airways. So if you've ever suffered from a chronic sinus infection or sinus infection in general, you know what I'm talking about. And this reduces epithelial barrier function and also leads to the increased potential for chronic infection. And then once those infections are established, it becomes very difficult to treat. And just as a fun fact, sinusitis accounts for about 1 in 10 doctor's office visits for which an antibiotic is prescribed. This really contributes to our overuse of antibiotics in treating sinus disease. So we feel that if we can better understand or get a better handle on the ecology of chronic sinus infection, this may go some way in reducing the number of antibiotics that we need to use to knock out some of these chronic airway diseases. In the case of CRS, or chronic sinusitis, Staph aureus is generally regarded as the canonical pathogen. It's the one that's most frequently isolated from CRS sinuses. But interestingly, Staph also colonizes the nasal cavities of about 50% of individuals, but asymptomatically. So it doesn't appear to be pathogenic in a lot of the population. Interestingly, chronic sinus disease is typically established by the same strains that were previously carried asymptomatically. So it's not just the case of that a new strain of Staph is coming in and driving infection. So this kind of raised the question for our lab is what host and microbial factors tip the balance between Staph just living as a commensal in the upper airways, but also what determines whether it's pathogenic in the sinuses, and can we start to understand the factors that feed into this sort of dual lifestyle that Staph takes on. So we asked many of the similar questions that we did about Pseudomonas, but in this case we focus on Staph. How is its pathogenicity shaped by its co-colonizers? We're also able to sequence sinus mucus that we get from patients treated here at the University of Minnesota. But the nice thing about sinus disease is that we're also able to sample healthy controls. So these are individuals going into the operating room, being treated for facial reconstruction, septal deviations, pituitary tumors. So we can sample their upper airway microbiota as well. And from these sequence data, each column is an individual, and the density of the bar indicates the abundance of each bacterial genus within those individuals. What these data tell us is that while both non-CRS and CRS patients share many of the same organisms, there's clearly a shift in average relative abundance. And that includes an increased abundance of oral-associated microbiota, including Streptococcus, Prevotella, and Fusobacterium, that once again are adept at degrading mucin. And so we asked, in the context of chronic sinus disease, could mucin be supporting the abundance of these and other low abundance anaerobic organisms in CRS? And are these feeding into the behavior of some of the more traditional pathogens that we find there as well? So these were some data generated by a former PhD student, Sarah Lucas, who is about to start her own independent lab at the University of Syracuse in New York. And she performed many of the similar growth experiments that we did with Pseudomonas a number of years earlier. What I'm showing here on the left-hand side, this bottom bar, this is the ability of Staph aureus to grow on mucin as a sole carbon source. So similarly, it does a very poor job. But when Sarah started to amend that medium with other metabolites, including lactate, case amino acids, and glucose, which we view as representative of a mucin degradation process, Staph started to grow to a more appreciable optical density. But interestingly, if she performed these experiments under anaerobic conditions, so fermentative conditions for Staph, well, we do see a significant increase in bacterial density in the media where she started to supplement them with other metabolites. They still don't really get to an appreciable optical density. So this suggests that aerobic respiratory conditions are required for Staph to utilize some of these metabolites that are representative of the mucin degradation process. So then Sarah wanted to perform her own enrichment experiments. So she went to our Department of Otolaryngology and got sinus samples during surgery. And then she performed that enrichment on our defined mucin medium. So these are 8 patients that she sampled. And this is showing the abundance of each bacterial taxa before enrichment and after. And what I'm showing here on the right-hand side is the aggregate data once again. And I hope you can see that in this enrichment, we're selecting against Staph aureus. Again, we didn't provide an electron acceptor for Staph to grow. So it allowed us to enrich all of these other organisms that we're interested in: Streptococcus, Legionella, Prevotella, Fusobacterium, and others. She then took those supernatants from the enrichment and again grew Staph on the mucin degradation byproducts. And what I'm showing here on the right-hand side relative to the black bar here, or the black line, so this is just mucin, intact mucin medium alone, that each enriched bacterial community was able to support the growth of Staph, even though there was less nutritional content available in the medium because it was removed during this enrichment process. There were 2 communities, including the yellow one and pink lines, that seemed to induce sort of a delayed lag phase and didn't really stimulate an increased culture density. And I'll get back to this phenotype in a couple of slides because we find it pretty interesting. So in addition to growth, Sarah also wanted to look at the transcriptomic profile of Staph aureus under these conditions. So here I'm showing similarities between communities. So regardless of what the composition of the community that Staph was cultured with, I'm showing similarities in terms of which genes were highly expressed. One of the most interesting ones was the NAN operon, which is involved in sialic acid metabolism. So again, this is one of the sugars that decorates those oligoglycans on mucin. And what these data would suggest is that that Staph, which can't grow very well, doesn't induce its sialic acid metabolism genes when growing on its own in the presence of mucin. It can use sialic acids when they're cleaved or provided in trans by some of the other organisms that it's growing with. We also found a number of genes involved in amino acid catabolism, many of which converge on the utilization of glutamate. Now, these genes have previously shown to be important in staphylococcal infection, particularly in skin and soft tissue infections. So this might suggest that Staph is also likely using amino acids in their catabolism as well. And then, like in the case of Pseudomonas, we also see an increased expression of ACSA involved in the conversion of acetate to acetyl-CoA. So what these data tell us is that acetate, one of the primary fermentation metabolites of anaerobic mucin degradation, are also being used by Staph in this cross-feeding relationship, regardless of the community composition that they're growing with. We also found many differences. So here I'm taking 3 communities, enrichment 1, 2, and 3. So these are the 3 communities that stimulated the greatest amount of Staph growth when their supernatants were used to culture Staph. And this is just a heat map of a number of genes that we find differentially expressed, and these are dependent on the specific composition of the communities that Staph is growing with. So on the right-hand side here, I'm showing that in this case, this particular community, enrichment 1, stimulates the expression of a number of genes associated with acute virulence and biofilm suppression. So this includes the AGR regulon, which is the master regulator of virulence in Staph, but also delta hemolysin, immunodominant antigen B, staphopaine, and also repression of the ICA adhesin gene. So these are thought to be involved in Staph aureus biofilm formation. So this suggests that one particular community may be stimulating genes associated with virulence and reduced biofilm growth. But on the flip side, 2 of the other communities that we co-cultured Staph with seem to be increasing the expression of ICA adhesin genes, including the Holin protein side A. So these are well characterized for their role in Staph aureus biofilm formation. And yet these 2 other bacterial communities are also inducing genes involved in metabolic stress and nutrient limitation during infections. This includes lactose and galactose metabolism, sulfate starvation, the methionine biosynthetic pathway, and also branched chain amino acid synthesis genes. What these data might suggest is that Staph, that these communities may be promoting Staph resistance to oxidative and nitrosative stress in vivo, which tells us that some cross-feeding relationships may affect Staph persistence and tolerance to host-derived stress that they encounter in the airway. So this was a pretty exciting result for us because very clearly Staph behavior depends on the other bacteria that are colonizing the sinus cavity in vivo. But at this time, when we were doing these experiments, we didn't really have a great handle on which bacteria are driving these interactions. But we are starting to get some clues, and we're currently following up on these clues. So I have a recent postdoc, Josh Fletcher, who's still in the lab, and he made the observation that appeared to be In our CRS patients in particular, this anti-correlation between Staph aureus and two other bacteria, Prevotella and Fusobacterium nucleatum. If we plot those data on this dot plot here I'm showing on the right, there seems to be this bimodal distribution in terms of bacterial abundance. So this is the percent relative abundance of each bacterium. So there appears to be this anti-correlation between the two. Interestingly, the two communities that I showed you earlier in the yellow and pink, they had two of the most abundant organisms in those communities were Prevotella and Fusobacterium. So they might be repressing staph growth. And so to test this in more detail, Josh has recently been doing some direct co-cultures between Fusobacterium and staph. In black, I'm just showing staph growth on a nutrient-rich medium, Brucella broth, and it grows just fine. But when we supplement that medium with either Fusobacterium nucleatum supernatans, or two of the short-chain fatty acids that it produces, propionate and butyrate, we do see this induction of a delayed lag phase. So very clearly, Fusobacterium is creating metabolites that's at least early on inhibiting staph growth, suggesting that there might be more to this antagonistic interaction. And Josh is currently following up on this in more detail, and we will have a publication coming out that dives into the mechanism behind this antagonism in the near future. Kalai, how much time we got?
You have another 10.
10? Okay.
Yeah, 10 to 15 minutes. We
Okay.
stop. Yeah.
All right. So one of the most exciting areas of our lab's research now is, we feel we have a pretty good handle on the way that some of these anaerobic microbiota are affecting more traditional pathogens that we find in chronic airway disease. But really, by comparison, we know very little about the role or the effect of some of these commensal bacteria on the host itself, at least in the context of the airways. A lot of this has to do with the lack of compatible model systems that allow us to co-culture airway epithelial cells with some of these anaerobic organisms. So these bacteria need oxygen, whereas these bacteria don't particularly like oxygen, whereas if you want to culture host epithelial cells in the lab, you require an oxygenated environment. And so this has really limited our ability to probe interactions between many of these anaerobic microbiota and the host itself. And so we wanted to come up with solutions to get around this problem. And so a former postdoc, Patrick Moore, came from the University of North Carolina, so he's an epithelial cell biologist. We tasked him with coming up with a model system that would allow co-culture of some of these anaerobic organisms and host epithelia, at least for a short amount of time, to allow us to probe these interactions in more detail. So to do so, he came up with a system in collaboration with Coy Laboratories, where he cultures airway epithelial cells as you would normally, just in normal tissue culture flasks, and then grow them on transwell plates, which is also conventionally used to culture polarized airway epithelium. And on the apical surface, they overproduce mucus. So this was an important consideration for us. And then once they're fully polarized, we then move them into our anaerobic chamber, where we grow them in this multi-well manifold that is very much like a transwell plate, except the base is oxygen permeable. So the plate can still hold cell culture growth media. But if we introduce oxygen from an external source and pump it into the bottom of our growth apparatus, the basolateral side of these airway epithelial cells remains oxygenated, where the apical surface is still exposed to the environment of our anaerobic chamber. So this should give many of the anaerobic organisms a chance to grow. And we started out using Calu-3 cells. So this is an adenocarcinoma cell line that are derived from goblet cells. But we use this as our starting point because they do overproduce mucus on the surface. And we know that at least when Pseudomonas colonizes, it tends to reside in the mucus itself as opposed to directly against the epithelium. And so this is why we wanted to use this cell line as our starting point to try to optimize this model system. So Patrick did some initial characterization of just the airway epithelial cells using this model. And so he cultured them in our dual oxic anoxic culture platform, which we refer to as DOAC for short. And then he looked at cell viability over time. So relative to these gray dots, which are Calu-3 cells grown normally under normoxic culture conditions, and relative to cells that are just put in our anaerobic chamber without given any oxygen, we do see the survival of airway epithelial cells after about 48 hours. About 72 hours, they start to die off, and this might be due to toxic metabolites in the media. but at least this gives us a period of time in which we can start to study interactions between anaerobes and these viable epithelial cells in our mixed oxygen culture conditions. He then measured transepithelial electrical resistance, and we compare this readout under fully oxygenated conditions relative to our mixed oxic conditions. There doesn't appear to be any significant change in tight junction formation. So this suggests that cells are fairly happy and intact. And then we've also measured cytotoxicity of a number of different cell lines and also primary cells as well. And these data are shown, this is after 48 hours. And so we already knew that we could culture Calu-3 cells, but RPMIs, this is a nasal epithelial cell line, so they stay viable over time. And also primary human tracheal bronchiepithelial cells do fairly well in this model as well. So once we were satisfied with this model, then we could start to treat with some of our anaerobic communities of interest. And in this case, we just started out with an enriched anaerobic community that we got from airway mucus. And we chose it just because it contained many of the organisms that we're interested in, including Prevotella, Streptococcus, Veillonella. And we felt it was fairly representative of an airway-associated anaerobic community that we culture in the laboratory. We can use microscopy to visualize colonization of these bacteria on the epithelial cell surface. And then we can use enumeration to quantify the viability of anaerobic bacteria when we use them in this co-culture system. So relative to the inoculum, after 48 hours, if we just use a PBS wash to collect anaerobes off the cell surface, we get a little bit of a reduction in bacterial load. So this was a little concerning. But then when we used a more aggressive wash to recover anaerobic bacteria from the surface, we do get about an order of magnitude increase in recovery. So this suggests that bacteria are not only viable, but also growing as well. And it might suggest that because it requires a detergent wash, they're either forming a very tight association with the epithelial cell surface, or they're being internalized, and we're not able to wash them off with PBS during our initial wash. Importantly, when we measure just our strict anaerobe inoculum or growth relative to the strict anaerobe inoculum, we also get increased recovery. So this tells us that at least the conditions that we're using under these experiments are sufficient to allow the recovery of these anaerobic organisms, suggesting that they're viable and happy under these conditions. When we measure cytotoxicity, this particular anaerobic community doesn't seem to be driving cytotoxicity or cell death, at least after, this was 24 hours of treatment, I believe. But when we use transcriptomics to look at the host gene expression profile after treatment, we do see the increased expression of a number of pro-inflammatory marker genes. This includes a number of cytokines and chemokines, superoxide dismutase, complement, ICAM1, and these are all markers of inflammation, suggesting that at least this bacterial community is being sensed by the airway epithelial cells, and this is also driving a pro-inflammatory response. Probably the most exciting experiments that we have ongoing now in the lab are combining this dual oxic-anoxic culture system with single cell RNA-seq profiling of airway epithelia in response to anaerobe challenge. So this, single cell RNA sequencing is being used widely in infectious disease research. But to my knowledge, typically relatively little is known how epithelial cells are responding at the single cell level to bacterial challenge. And so this is how we wanted to leverage our platform to get at this question in more detail. In this case, we again grow, in this case, it's primary cells under our mixed oxygen conditions. And then we treat it with Fusobacterium nucleatum. So again, this is an anaerobe associated with the oral cavity, but we find it in our sequence data. And it also has implications for a number of other body sites as well. So that's why we selected Fusobacterium as our starting point. We treat our primary airway epithelial cells that consist of all canonical airway cell types, including ciliated cells, ionocytes, secretory cells, and basal cells. After treatment, we homogenize our airway epithelial population, and then we use the 10x chromium controller to capture individual airway epithelial cells and use Illumina sequencing to characterize the transcriptional response of this epithelial cell population in response to a bacterial challenge relative to an untreated control. So for this, we use the Seurat-R package, and this was designed for single cell experimentation. And this allows us to cluster airway epithelial cells depending on their differential gene expression profile. So each dot on this UMAP plot is representative of a single cell, and they're distributed based on their global transcriptional profile. Using marker genes for each cell type within these clusters, this allows us to identify the individual cell types found within this distribution. And not only does this allow us to identify each cell type, it then allows us to perform differential gene expression between each cluster, accounting for both the effects of not only cell type, but also bacterial challenge as well. One of our first findings is when we're looking at host-specific gene expression, we do see a notable differential expression of pattern recognition receptors. So these are proteins responsible for the initial detection and response to bacterial pathogens. So throughout this plot, these heat maps are really just showing the distribution of gene expression of TLR1, TLR2, NOD2, and NLRP1. These are canonical pattern recognition receptors that are involved in the response to bacterial colonization. And what these are showing is that in the case of TLR1, TLR2, and NOD2, they appear to be preferentially expressed in secretory cells. But by comparison, NLRP1, which is responsible for the detection of intracellular DNA, we see here the preferential expression of this particular pattern recognition receptor in basal cells. And we also see this for 18 other pattern recognition receptors. So this might suggest that the response of airway epithelia is going to be heterogeneous in terms of cell type and how they're responding to challenge by a given bacterial pathogen. And that's exactly what we see. So in the case of basal ciliated cells and secretory cells, we see the differential expression of, this should be 55, 240, and 87 genes that vary between cell type. And I just want to make the point that this differential gene expression would be lost in just a bulk RNA sequencing experiment. So we're starting to get into really just at a single cell level, how different epithelial cell types are responding to challenge by Fusobacterium nucleatum and getting a better sense of how the host might be responding to this anaerobic organism. On the left-hand side, I'm showing a subset of genes that are differentially expressed that relate to inflammation. So we see here matrix metalloprotease is differentially expressed in basal cells, for example. We see chemokine CXCL17 also increased in expression after Fusobacterium challenge. And we also see this for a number of other genes in ciliated and secretory cells as well. But perhaps the most striking result is the range of genes or pathways that are involved in tumorigenesis. So all of these genes are associated with cancer or tumorigenic processes. And to our knowledge, Fusobacterium hasn't really been associated with airway cancers in the past. But we do see the increased expression of PIP, which is part of the phosphoinositol 3 kinase pathway. It's involved in a number of cancers. And more recently, Leopoldo Segal's group out of NYU published a paper that there's an association of airway microbiota composition and the upregulation of the PI3K pathway in lung cancer. And probably the strongest association between the expression of the PI3K pathway had to do with the abundance of Fusobacterium and Prevotella as well. So these data might start to be providing some mechanistic insight to how particular bacteria might be driving tumorigenic processes. It may also be that the airway epithelial cells are just trying to repair themselves after bacterial challenge. But at the end of the day, just gaining further insights in how the airway epithelium might be responding to bacterial challenge, not just anaerobes, but any bacterium when growing under oxygen limiting conditions is highly relevant to our understanding of airway disease. And so we're excited about this model. This paves the way for additional studies of other organisms that we're interested in, Prevotella, Bionella, for example. These organisms are also found at all kinds of sites of infection throughout the body, periodontitis, wounds, GI microbiota, the vaginal tract. And we feel strongly that this model sort of paves the way for understanding microbial dynamics across the body in the context of health and disease, really at any site where microbial ecology matters. So we can start to gain a better interest in host pathogen interactions and how this might be driving a variety of clinical conditions. So
2 minutes, yeah. Oops.
Perfect. 2 minutes. Good timing. With that, I'll stop there. I just acknowledge those that are responsible for the bulk of the work I just presented. Talia and Kayla are responsible for most of the bioinformatic analysis that went into the work. Jeff and Sarah were responsible for the mucus degradation in staph and Pseudomonas. Patrick was responsible for the epithelial cell culture model, and Sabrina is our analytical biochemist who's very skilled at mucus degradation analysis and metabolite quantification. So I'll stop there, and I'd be happy to take, it looks like, a number of questions that are in the chat.
Yes.
And thanks to everyone again for attending.
Thanks, Ryan, for the wonderful talk. I really learned a lot of new stuff. There's a couple of questions. I also have a question. The first one is from Igor Grekov. Did you check this growing in mucin as in a biofilm?
Is the question, so when
what happens if you grow Pseudomonas aeruginosa biofilm in mucin, basically?
Right, yeah. So we haven't looked at that ourselves. There's a number of groups, including Matt Parsek comes to mind, where if you have an artificial mucus medium, I think Marvin Whiteley's lab and Sophie Darsh and others have shown this too, that if you start to add mucus or mucins to the growth medium, that increases the viscosity and sort of creates some structure to the growth environment. The Pseudomonas does adopt a biofilm-like phenotype in the form of aggregates. We haven't really looked at that, but I suspect that they're likely going to be aggregating in similar ways when we grow them in our mucin medium. We haven't really looked. It's a good question.
Okay. So the next question comes from Giri Narasimhan. Does your work inform us on how to manipulate the composition and behavior of entire microbiomes and achieve desired composition profiles?
That's sort of the ultimate goal, right? I think we're just scratching the surface of understanding what makes a commensal versus pathogen. I think it's very context dependent. They can thrive asymptomatically and promote oral health, for example. But if they start to disseminate and show up at the wrong place at the wrong time, and the wrong relative abundances, and then that's when you start to run into problems. So I think the more we can understand about how these organisms are interacting with one another and the host and traditional pathogens, we can work towards that goal and start to manipulate the bacteria that we want to have around, as opposed to the ones that we don't.
Thank you. So here comes a question from Norman Fry. Dr. Fry says really interesting seminar. And he's interested to know if any antimicrobial treatment of CF patients is actually being used against the microbial community as opposed to single species.
Yeah, I
don't think so.
It's a loaded question. No, I don't think so. There are guidelines that our clinical labs need to follow to track antimicrobial susceptibility and the administration of our frontline antibiotics. I think we can. There's nothing stopping us from using more like broad spectrum antimicrobials to try to target more of those organisms. I think we run into the issue though that, particularly in the case of the CF lung, I like to think about this a lot, that because you have a genetic defect and a difficulty in clearing mucus, something is going to colonize that environment, whether it's Pseudomonas or something else. So I think if we can tip the balance and promote the growth of organisms that might not be as pathogenic, or sort of cultivate a healthier ecosystem, might go a long way in improving airway function, or at least limiting pathogenicity of that site of infection. There are some anaerobic specific antibiotics that can be used. Metronidazole is one that comes to mind. It's not widely used in the context of CF therapy, but there are options for direct targeting of some of the organisms that I just talked about.
So the question for you is that you've identified some specific microbes that are sort of helping the survival of Pseudomonas. How do these bacteria respond to the current treatment? Like, for example, we do tobramycin and lactam and aminoglycoside, whatever the current recommended treatment. How do Fusobacterium, Prevotella, all those respond to those specific antibiotics?
Right. It's a great question, a challenging one to get at. I would refer the audience to the work of Rob Quinn at Michigan State University. He's a good friend of mine. He's done a lot of work where he cultures whole bacterial communities in vitro and uses sort of a similar stratified system to see how each one is responding to drug treatment. He's seeing all kinds of interesting interactions, including those that survive versus those that don't. With patients, somewhat challenging to track, mainly because when you collect sputum, it's passing through the oral cavity. And so some salivary microbes might also be being collected in addition with sputum. So at the end of the day, it's somewhat challenging to figure out how many or which anaerobes and how many are coming with the sputum itself versus how many are coming from the oral cavity. That's another reason why we switched to the chronic sinus infection, because we don't need to sample through the mouth anymore. So yeah, it's a challenging question to get at.
But what if you do the experiments you did with even with epithelial cells with this various challenge? What if you challenge it with antibiotics?
Yeah.
Right.
Yep. Yeah, we could. We have not explored that yet.
Yeah.
It's definitely doable.
Right.
There's too many questions. Too many questions. Not enough time.
Well, you need a larger lab, more funding.
Yeah, exactly. Don't we all?
That's real, exactly. This is the other interesting question is, you have used the glucose as a control media to show growth, but, Pseudomonas preferentially uses other carbon sources like succinate, right? And also, so how does that would change? I don't know, just that why didn't you consider using succinate as your control rather than glucose?
That's a great question. Yeah. So yeah, succinate would be another good control in that case. So when we did our initial targeted analysis of mucin-derived metabolites, we couldn't detect succinate. That's a limitation of our instrumentation. So we didn't include it initially in our cross-feeding model. Yeah, I mean, Pseudomonas does like succinate a lot more than glucose. Not sure about staph, but
yeah. So the other interesting thing I noticed is that, do you think Pseudomonas uses preferentially the glycosylate pathway rather than going through the whole TCA cycle, or does it need something to trigger the other pathway?
Yeah, I'm not a metabolism person, so I don't really know the answer to that. It makes my head hurt. I don't know. I honestly don't know.
Because the Pseudomonas does have very defined system to take the system through alpha kg to the other realm, right? So it's kind of interesting question. But anyways, so we are at the tail end of the hour, 10:30, and so I think we will wrap up today. So I would really like to thank Dr. Rick Hunter. So Dr. Ryan Hunter for a fascinating journey through his research and things that, going from multicellular to multibacterial to single cell analysis, it's really going to change the way we think about infections and immune system. And we didn't have a time to get to a question I have now these days is that we need to be treating or trying to figure out how to take care of our immune system, because ultimately the death and destruction is caused by overacting immune system.
Right.
So we need to sort of move in that direction rather than single pathogen, single treatment is like microbiome and immune system is where we need to go.
Yep.
With that, I'd like to thank the audience for attending. A reminder that a link of the recording will be emailed to you and will be available to view for 7 days. The next session in the JMM seminar series will take place on June 30th at the same time. The lecture will be delivered by Dr. Andrew Broadbent from the Department of Animal and Avian Sciences at the University of Maryland, College Park. The title of his talk will be Understanding the Pathogenesis of Avian Viruses with the View to Improving Control. And I wish you all a happy weekend and happy Memorial Day. And see you all soon at the next seminar series. Once again, thank you for participating. I hope some of you noted there is an anaerobe conference that organized by Microbiology Society. We hope to see some of you there. Thank you again. Thank you, Dr. Ryan.
Thanks, Kalai. Thanks, everybody.